that cause the common cold. And so is coronaviruses, as far as we knew before. So when you look at this little table, you can see that they were not considered to be very severe. And they mostly are something that register in children because they get them for the first time. What happens then during your lifetime is that you build up immunity. And that's true for coronaviruses, adenoviruses, flu to some extent, although flu, influenza, tends to change quite a lot. And there are new variants coming every season. So that's why influenza is always a serious problem. But if you look at this, it does not look like coronaviruses were particularly dangerous for adults and the elderly population. Of course, this is completely different for a new virus. So once a new pandemic comes into the human population, we are all potentially susceptible. And then we're getting used to seeing these sort of pictures. This is actually taken from our lab here at KI, where the PhD students work under high biosecurity with this virus. So when a new coronavirus comes into the population, which we experienced in 2003 for SARS-1 and more recently for MERS in the Middle East, there was a completely different situation. People get quite sick, the virus spreads, the viruses have pandemic potential. Luckily for these two viruses, things got under control relatively quickly. But for this virus, it's been different, and it spread very rapidly around the world. SARS-CoV-2 is probably a little bit less pathogenic, but more contagious compared to SARS-CoV-1. And the numbers are very, very high, more than 25 million recorded cases, and that's only the ones that have been measured, and now over 900,000 deaths around the world. So this is a completely different situation than the previous coronaviruses. And this is because there was no immunity in the population. So the problem really is a question of immune responses and whether you can fight this with strong adaptive immune responses. And what's clear when you have a new coronavirus is that the elderly are highly susceptible, and the children and young people are actually less susceptible. So there is a clear correlation with disease severity and age when you have a new pandemic viruses like this. What the whole world has been trying to understand is how we develop antibody responses to this virus. Antibodies are soluble molecules. They are circulating in our blood. They're produced all the time. They're produced by B cells, and B cells are also produced all the time. We are making new B cells all the time, and they have the potential to recognize any foreign molecule that we might encounter. So this picture just shows the small little Y-shaped antibodies binding to a virus particle. And what's important then in the case of a new pandemic is to understand what sort of levels do people develop in terms of the antibodies, and what sort of quality do these antibodies have. And I'm going to show you some actual sort of real unpublished data from our laboratory just to illustrate a little bit about how the antibody response actually looks when you start looking at lots of people. And this is of course important for political decisions as well, because every country needs to understand what is the spread of the virus, what is the seroprevalence in the population. It's also important to understand how long will this last? What's the durability of these responses? Are we protected for months, for years, for the whole life? So these are really important questions to understand. And fourthly, the level of immunity that we get from the natural infection is some sort of benchmark to what you want to achieve with the vaccine. And in some cases, you develop good immunity with a natural infection. In some cases, you get a better immunity with a vaccine. This just depends on what kind of virus we're talking about. But these are central questions. And I think every country around the world have been dealing with these questions over the last 6 months. Here in Sweden, there are, as in many places, a lot of commercial companies rapidly develop tests. And there were many different reports in the public press. What's the seroprevalence of Sweden, of Stockholm, many different numbers circulating. And it was quite unclear for some time. And now it's settling down, and the numbers are much, much more sort of solid. But what's tricky here is, of course, that the tests are not all perfect. So it depends a little bit which test you're using and what sort of population you're studying. And if you are doing an unbiased testing, or if it's people who seek testing, and they usually represent people that actually want to be tested because they had symptoms. So these are all considerations when you talk about percentages. I'm going to come back to this with some actual data. What's important when you measure antibodies is to measure against a virus component that you can produce and make a very reproducible test. And if we now think about this virus, they're tiny, of course, viruses, but they have this characteristic that they have these spikes on the surface. And the spike for this virus, and this is true for many viruses, in this case, are really protruding from the virus particles. You can see it on electron microscopy. And that's the molecule on the virus that will bind the cell that it's going to infect. So it's interacting with the cellular receptor. That's exactly the interaction you want your antibodies to stop. Because if you have antibodies that can block that interaction, you have protective immunity. So these little spike molecules are central. They are the targets of neutralizing antibodies, and that's what you want to elicit with a vaccine. And here I'm showing the particle. This is actually the real structure of the spike that I sort of pasted in here. So this structure atomic resolution was published already in this winter, very, very soon after the virus was discovered. And based on that, the whole world could now make these proteins for different binding assays. And that was published in Science. This is exactly the molecule I'm talking about, the spike. We are making it in our lab and produced here by postdoc Leo Hankey. And it's central for detecting antibodies and for understanding how antibodies might block the infection. I'm just going to show you a little bit of data. And just to give you an idea, we very early on collaborated with clinicians to collect samples from patients that either had a mild or more severe or a very severe infection. And these are just some of the groups that we studied. And to show you, this is about 100 people, 105. All of these people develop really well detectable antibodies using an assay that measures antibodies against the spike protein. So this is how it looks. And you can see that there is a range of magnitude of the response. And this, from the highest to the lowest here, there's actually about 1,000-fold difference in titers of antibodies. So everybody develops antibodies in this group of people, but the levels vary. All of these people had confirmed PCR, that is virus test. So they were all confirmed to have had the infection, and they all develop antibodies. We looked more closely if it matters if you have a mild or a moderate or severe infection, and divided up these individuals in 3 categories. And it was quite clear that the more severe symptoms, the higher antibodies levels you have. Here we are looking at 3 different subclasses of antibodies. IgG is the most commonly measured, and the antibody subclass that will last the longest. It stays around the longest in the blood. But IgA, for example, is very important because it's produced and made by cells in the mucosa. So that's where the virus is, in the lung tissue or the respiratory tract. And there is a lot more IgA in people with severe disease. So this was quite clear. And if we were interested in what happens if we now screen people that we don't know if they've had the infection. And we simply collected serum from blood donors, healthy blood donors, and pregnant women, because there is a collection weekly from these 2 categories of people at the hospital here. And then you can see that you pick up very clearly who's been infected, and many of these people did not know that they had been infected. So this is actually a good sort of indicator of seroprevalence in the population. And but you can also see from the levels here that they are slightly lower. Most of these individuals had a mild, perhaps completely asymptomatic infection, so their antibody levels were a bit lower. But there are clearly some individuals here that are very clearly exposed. we were actually looking over the weeks during the winter and the spring and the beginning of the summer. And the raw data is difficult to see here, perhaps, but we use a very sort of carefully sort of cutoffs for how we determine who is positive and not. And the curve that you can see is really showing how antibody immunity in Stockholm develops over this period of time. And we could say that in the beginning of the summer, Stockholm was at 7%. Now it's approaching 12%. And I think that's where it is. But it's slowly plateauing out because there's less virus spreading. And this is just an example from here, from locally, but very many different countries are reporting similar sort of percentages now. And even New York, that had a very bad pandemic, their top seroprevalence level, even in the worst parts of the city, were 15%. And if you look at all of Sweden, it's lower. It's about a few percent, and all of Spain, it's 5%. So really, we are not approaching herd immunity, and no country is. And this is something we did not know in the beginning. It was not known if this virus would induce herd immunity. But what we can see is that we're not reaching those levels. Another question that's been discussed a lot lately is, do people lose their antibodies? Are they short-lived? And because there were surprisingly low numbers of people with antibodies, this question came up. And there was a concern that you have such low levels and that they're so short-lived that you actually lose people and they're only positive for a few months. That does not look like it's the case. The antibody response is fairly stable, but it will depend on the levels you start from. So if you start high, you will, of course, have a much more sustained response. But even if antibodies do decline, it's not a big concern because the cells that make the antibodies, that are called memory B cells, they persist. And memory B cells, this is how the immune system is so cleverly built up, because these cells are programmed to live for a long time, years, and they circulate, and they don't produce antibodies anymore because they're sort of quiescent and quiet. They're just circulating, waiting to see if there's new signs of infection. And if there is a re-exposure and the same virus is coming into your body again, then poof, they start making antibodies again. And this is a process that's quite well characterized, and we can measure these cells in the lab, and we can see exactly which memory B cells become antibody-producing cells against coronavirus. So it's not an assay that you can do on large scale, and it's not something that you can do sort of at the population level. It's a laboratory-based assay, but in 100% of the cases that we've looked, people that had antibodies have memory B cells. And I think this is sort of a reassuring to say that once you've been exposed, you actually have some level of immunity that would last a long time. And I think understanding the cells and how these antibodies are made is quite fundamental for understanding how immunity works and how vaccines should work. So a question that's come up is, if so few people have antibodies, why is the pandemic now decreasing in this country? Because it is. There's very few cases now, very, very few cases even in Stockholm. So what is the basis for this if we don't have herd immunity? And there's a few possibilities. One that I probably believe in the most is that our public health measures have actually been effective, even if we've had more relaxed public health measures than many other countries. It does make a difference if you take these rather simple measures like social distancing and hygiene. That's, I think, paying off. Another thing is that there could be individuals that are quite resistant and fight this virus with natural barriers and have very, very transient infections so that they do not develop adaptive immunity. And that's, I think, not unusual for viruses, that you have most healthy people have good barriers to control the virus early. And a third possibility that many people are researching at the moment is if there is some level of cross-reactivity between the benign circulating coronaviruses that we knew existed before and this coronavirus. That could be a very, very small fraction. It's probably not accounting for a big fraction. But there are There are certain other possibilities here. There is some degree of T cells. The virus might be changing and becoming a little bit more benign. So there's probably a combination of all of these things that make the pandemic much sort of reduced at the moment here. Vaccines, just a few quick words about vaccines. Vaccines will be needed to induce herd immunity, and we know that the natural infection will not achieve that. So if we really want to have a kind of a situation where everybody have good protective antibodies, we should develop a very good vaccine. And what we know about vaccines in general is that neutralizing antibodies that I spoke about, the kinds of antibodies that can block infection, bind these spike proteins, they are the reason our existing vaccines work. So I have a long list of viruses there. They all, the vaccines against all of these viruses mediate protection because they make these antibodies. So it's quite clear that the correlate of immunity for successful vaccines is antibodies. And when it comes to the different types of vaccines that are being made at the moment, I think it was mentioned here previously, some vaccines, like for yellow fever, for example, is a rather older vaccine. It's made by making the virus weak and therefore safe, but good, but sort of still enough to replicate a little bit so you get an immune response. They're called attenuated vaccines. And our measles, rubella, mumps vaccines are also that kind. There's some vaccines that are inactivated virus particles, where you kill the virus with chemicals. And there are some examples there of vaccines that are made in that way. More modern vaccines are based on gene technology, and that's our papillomavirus vaccine, our hepatitis B vaccine. And they are also highly effective. And that's what industry is focusing quite a lot on, because they can be made in a very reproducible way. There's even more sort of new platforms, like in encoding the virus genes of interest, in this case coronavirus spike protein, in a DNA or an RNA vector, or in another virus, a common cold virus, for example, so that you let the body produce the antigen, the protein, to which you want an immune response. All of these platforms are being explored for COVID-2, except the first one. Well, that might be as well, but it's nothing that's very progressed. But all of the other ones are already in far progressed phase 2 and phase 3 trials. And you will have heard about some of these. You can see here on the list what kinds of vaccines they are. The last two categories are experimental. We don't have vaccines that work that way yet, but they're being pursued. And I think we will see from these trials what's going to be most effective at eliciting a good antibody response. I have an acknowledgement slide, which is my lab and our collaborators, and I'm happy to take questions.
So much. Give her a warm hand to Gunilla. So I want to ask the first question. You're a member of the Nobel Assembly that actually picks the medicine Nobel laureate for this year. Can you tell us who it's going to be this year?
Nice try.
Nice try, okay. Is it hard to pick who will get the prize?
Yes, it is, because there's a lot of different areas of medicine to cover, and there's a lot of important work being done. So it's a matter of really identifying what's a very distinct discovery and what's important and novel.
Yeah. So within 4 weeks, we will know who actually gets it. So we'll have to wait. Okay. So now it's time for some questions from the audience. Do we have any questions here in the room first? Some teacher want to ask a question? Yes. Yes, the microphone. So your name first, maybe?
Rachel Harris. You talked about how the memory B cells would stay in the body. Would you still get sick again, or would those cells counteract the virus before you would develop symptoms?
I do think that most of the time, most of the vaccines that we have are actually working by reactivating these memory B cells, and it's pretty rapid. So within a day or two, they become antibody-producing cells. There's also some level of circulating antibodies from long-lived antibody-producing cells that I didn't really talk about here, but some circulating pre-existing antibodies together with the memory is what gives us protection, and I think it happens quick enough to stop the virus at the gate. So the sooner you stop it, of course, the better, because otherwise you will establish low levels of infection, and then you could be a spreader, and if you really want to stop it as effectively as possible.
Okay, thank you. So you had also a question, yeah?
Hi, my name is Nicole, and my question was, there's been a lot of speculation and debate about what a second wave might be, what that is, and what does it mean for us globally, and I was wondering if you could talk about that, and even if it means more specifically, what does it mean for us here in Stockholm?
Can we just switch so you can stand there?
I think that we still are, we have to be humble that we don't know what's going to happen, and we have to continue with the precautions, that's for sure. I think we should be, of course, I think what we're seeing from having kept the schools open suggests that that is not the major sort of source of spreading the infection. So all of these incredible measures that Annika alluded to in the beginning, also that teachers are now finding different ways to teach, perhaps having half of the kids there and alternating so it's not quite as crowded. All of those things are important and have, it's sort of been, now it's getting almost like second nature that one can employ those kind of measures. It's definitely an extra challenge, but I would sort of be cautiously optimistic at this point.
Thank you, Gunilla. And I think, Pontus, do you have some questions that were sent in by the teachers?
Yes, I have, and I will choose this one. Do you think that the vaccine interventions will remain effective over time, keeping in mind that the viral mutation pattern of SARS-CoV-2?
So this is a question that relates to if the virus is slowly changing. And it is a virus with an RNA genome, and that is, of course, always a risk. But it does not look like this virus is changing extremely rapidly. And that's typical of coronaviruses. It's not a major concern as far as we know right now. There are some different variants circulating in different parts of the world, but it's not affecting how sensitive these viruses are to antibodies and to the antibodies that we know are neutralizing. So something to monitor for sure, important, but not a major concern at this point.
So I was thinking when I listened to you, I want to listen to this once again, because you said so many interesting things, but I didn't quite understand it all. But luckily, we have a TV team here from the Swedish National Educational Television, called UR, and they are recording everything. So we will also send out links so you can watch this, maybe show it to your students in your class. So we will send it out to you as soon as it's ready. So thank you so much, Gunilla, for this lecture. Give Gunilla a warm hand.